All right, well, it's a pleasure to be here, and it's great to follow the three terrific speakers we've had so far. I've really learned a lot. So I'm looking forward to telling you a little bit about some of the things we're doing. So I work at a place called the Institute for Systems Biology, which is a nonprofit research institute. It's affiliated with Providence St. Joseph Health, which, if you're not aware, is actually now the third largest nonprofit healthcare system in the country. So that's kind of our home base. And then I will talk a little bit about some of the things that were discussed here. So in terms of disclosures, obligatory ones here, the primary ones, as was mentioned, I'm co-founder of a company called Airvail, which I will talk about a little today because organizers wanted to hear a little bit about some of this in the real world and what's going on. I'm also on the scientific advisory board for a personalized nutrition company called Habit, and I'll say a little bit about it as well in terms of practical kinds of approaches. But first I want to talk about science, since that's what I do full time. So basically, we're all very familiar with this, right? That nutrition has a large number of health effects, and you can see here all kinds of different foods that have been shown to be protective against cancer. Now, for people in this room, they're not going to be surprised when I reveal the other side, which is that they've all been shown to cause cancer as well. And so, right. So in terms of nutrition health effects, they're complex. There's a need for personalization. There's a need for understanding. We heard wonderful description in Dr. Brannon's talk at the beginning of the lecture today about these complexities and just how intensive it is when you start thinking about the molecular detail that's involved in how your body processes food and how that's related to disease. So there's enormous complexity here to understand. And so there are many different systems biology inputs to this, many different ways that we can think about it. We heard a lot from Doug's talk that he just gave, which was great in terms of mitochondria and impacts, and there's many, many different kinds of elements that can be delved into here, and that's one of the things we're trying to get at. Now, if you think about the determinants of health in the United States, this comes from a New England Journal of Medicine paper in 2007. It's been estimated that about 30% of a person's lifetime health is attributable back to the genetic hand that they were dealt. About 60% comes back to behavior and environment, nutrition obviously being a huge element of all of that. And it turns out that only about 10% of a person's lifetime health is attributable back to the healthcare system directly. And so there's a huge need, obviously, to focus on the 90%. So the way we think about this is that you have a healthcare industry that is closing in on $4 trillion a year in the United States, a pretty staggering number. We also have a very large wellness industry. It's already been alluded to here today that the wellness industry has a mixed reputation, let's say. There's a ton of great actors. Nutrition is, I think, foremost amongst those in many, many cases. And then there's also a lot of, let's say, not very scientifically based approaches that are sold very broadly in the wellness space. So at least in our terminology, we think about this as being a rise of something we call scientific wellness. And this is what we push really heavily, especially in talks to the medical community. One of the elements that I think will resonate in this audience that's very obvious is that if you talk to typical physicians, how much nutrition training is there for a physician? And the answer is about 2 hours, and so it's about an afternoon or something like this. And so there's a huge need in our mind, certainly, for having what we call scientific wellness approach, which is really trying to predict and prevent disease before it happens, and to focus on optimization of health in the individual and enhancing resilience and so forth. So that's a big area for us. So in thinking about this concept, Lee Hood and I, and if you don't know, Lee's a very well-known biologist. Lee was the guy that invented the automated DNA sequencer that made the Human Genome Project possible. Lee is a member, one I think of 15 people who's a member of all 3 of the national academies and so forth. And so Lee has really this wonderful history in science. But Lee and I announced to the world a couple years ago a project that we were thinking about that we thought would help us to launch scientific wellness. And we called this project the 100K Wellness Project. And the initial vision for this was really to get 100,000 individuals and to get dense information on them. So this would be things like genomics, proteomics, metabolomics, microbiomes, clinical chemistries, wearable devices, and so forth. And it had one major goal, which was to get a data set that would be dense enough in terms of all the different kinds of information that would be available, dynamic enough so that we could watch people over time, so that we could see the early warning signs for all the major human diseases with the goal of developing ways to predict and prevent them. And so this has been really a driving force vision for all of that we've done over the last few years. And so to start this, what we did was to carry out what we call the Pioneer 100 project. So this was the first, turned out to be 108 individuals. And basically, what we did was to take them through a program like this so that we could test whether or not we could get all these different types of data, prototype this, so it was really a feasibility study. And this was all published in Nature Biotechnology a couple of months ago. And so, in essence, what we did here then was to carry out a longitudinal 9-month study, in this case, where individuals would come in and we would take them through a program where they would get all these data types measured from them, and then we would pair them with coaches, and then we had 3 different rounds. So there was round 1, round 2, round 3. And in essence, what we were generating was, first, we got whole genome sequence data on everybody, just to provide a baseline for analysis. We did a lot of different labs. So this was about 150 different clinical lab tests, about 700 metabolites and 400 proteins for every individual, again, 3 times during the year. We did gut microbiome, again, 3 times during the year. And we had them wear wearable devices, so Fitbits, things like this, so that we could get self-tracking and lifestyle monitoring. We would mine these information. This pulls data from well over 1,000 different published studies where, for interventions and so forth, but a database of actionable possibilities that will grow over time. And then what we did with these is that this creates what we call personal, dense, dynamic data clouds. Personal in the sense that they're for the individual, dense because they have a lot of information in them, dynamic because they change over time. And so in the longer run, what we're interested in is to gather all this information so that we can mine it to make new health discoveries that we can then return back to individuals after they've gone through proper regulatory approvals and so forth. As I mentioned, this all went through a wellness coach. This was also alluded to in the talks before, how critical behavior change is in all of this. Behavior change is extremely hard, as you know. Sandy Kaplan, who was, I will say, by far the most popular person on our study. People really love Sandy. But she's trained in behavioral coaching. We also had a study physician, Craig Keebler. So if anything became medically relevant, this would be referred back to the healthcare system. And so essentially it was really trying to focus on how do you take these data, make it possible, actionable for the person in the moment. And what's really essential about that is that we think this kind of focus on scientific wellness, making those data relevant to the person now, is really the key to having them stay in the study or stay with us or let us get this kind of data longitudinally so that we can really understand what's happening. So in that sense, in our mind, scientific wellness then is really the key to understanding disease because it's the way to understand the very earliest transition points from wellness to disease. And so that's what we're really keyed in on. So in terms of the initial study, this was, we did clinical lab discovery. We saw improvements on a number of different clinical markers. I won't belabor them. But this is essentially just using what are now standard clinical ranges. And when we looked at the individuals, the number of individuals who were out of range at baseline, and then how did that improve over time. So we saw a 21% improvement in terms of those markers for nutrition, 33% for diabetes, 12% for inflammation, 6% for cardiovascular, which changed more slowly. Incidentally, for those who have stayed with this kind of program by going through Airvail, this is now increased to a 20% improvement for those individuals. So it just sort of changes more slowly. We also did monitor a number of things about diet. This is just a very simple example out of the 9 months. One individual that came through, this person's come out publicly, so I can say who they are. This is Ed Lazowska, who's the Bill Gates Chair for Computer Science at University of Washington. Anyway, he came out with high mercury levels, was one of the things that popped up out of the study. It turned out by lifestyle he had really likes tuna sushi, was eating it about 3 times a week. That, of course, inflated his mercury levels. He then switched to salmon sushi, and over the course of 3 months, you can see that the amount of mercury in his blood went to half. Perhaps not surprisingly, is now totally normalized because he's stayed with us for the next year. And so, anyway, a number of cases like this. There were 2 other individuals that had this same issue. One of them was, again, for a love of tuna sushi. The other was for feelings in their mouth and so forth. So just kind of a simple example of the kind of things that you just find when you just screen a population. But what we really wanted to do was to look at these multi-omic diverse data sets, and that's really what the focus here was on. So here, these are just under 4,000 correlations that we found that cross different data types. So these are just looking at associations that we find between, for example, microbiome and metabolites in your blood, or metabolites in your blood and proteins in your blood, or proteins and lifestyle factors, or metabolites and genetic risk scores, and so forth. And so what I want you to think about just momentarily is that these data types had never before been measured simultaneously on a population of people. So this is the first time that this has ever been done to measure all of these things and just to start looking at the interrelationships between them. So as we start thinking about that huge complexity between diet and genome and disease and wellness and so forth, this is just the very tip of the iceberg in terms of understanding how all these things interrelate. It's funny because sometimes I get pushback on this, especially from certain people in medicine. We're like, oh, you shouldn't measure all these things, or we want to minimize the amount that we measure on every single individual. And I just couldn't disagree with that more. I think it's the notion that we will never make all of these kind of measures on sets of individuals to understand interconnectivity, I think there's just no chance that won't happen. And so the other thing that I think is important is to separate out what we do in terms of discovery, trying to understand how the human body is really interconnected and how do we understand these systems, from how we deploy certain things in economic relationships throughout populations as we go forward. That's very different kinds of rationale there, obviously. But this was kind of the basis for this initial study. So you can go through this, and in the interest of time, since it's a short talk, I'm not going to go through any of the details, but you can basically modularize these networks down into subsets where you have high interconnectivity with the subset and less so with the rest. And so you can build all these different modules for study. I'll focus on just one of these very quickly, which is, you can see here a relationship here between cystine, which is the dimer form of cysteine in the blood, and inflammatory bowel disease. And so what's interesting here is that this is an example that we found through this particular study, where you can take not the disease itself, but actually just a person's genetic risk for the disease, and relate that back into differences that you can see in nutrients in the blood. So in this particular case was quite interesting. So we found this negative correlation that said the higher your on average, the higher your genetic risk score was for inflammatory bowel disease, the lower the amount of cystine that you would see in that person's blood. Now, there's some interesting things that come out of that. One is that we then looked at different case control studies on the actual disease IBD. And it turns out that cystine in the blood comes out as one of the best candidate biomarkers in these studies. It's very different in individuals that have IBD. But what this said that was interesting is that we found that difference totally pre-existing in individuals ranging in age from their 20s through 89 plus, and basically through that entire span in which nobody has the disease, we already find this difference. So what that says is that that deficiency, if we want to call it that, in cystine is pre-existing. Now, one of the interesting things is that an interesting hypothesis comes out because cystine, sits upstream of the synthesis of glutathione, which is what combats oxidative stress in the body. We heard a great talk on oxidative stress just a moment ago around the mitochondria. But basically, one of the interesting elements here is that depressed cystine can actually lead to an inability to process as efficiently oxidative stress. So it immediately suggests a potential mechanism or a potential reason why these genetics are associated with increased risk for IBD. Right? Could be because of this depressed ability to combat oxidative stress. Since oxidative stress is a trigger for IBD, this means that over the decades of a person's life, they're more likely to go past their reservoirs of resiliency, transition into some kind of a disease state. Now, if that's true, what's really fascinating is that means you can take a person who's at high genetic risk for IBD and potentially, by shifting their nutrition, change them to someone who's at low actual risk for the disease. Now, this particular example we need to work out in more detail, all right? That's a hypothesis for now. But in general, this kind of thing basically has to be true. We call this the manifestation of genetic risk in the body. Gene variants don't mean anything in a test tube. They mean something in the body. So those genetic variants have to manifest somehow. And if you're using something like these dense dynamic personal data clouds, you're increasing your chances of being able to see what those differences, what those reflections are. And that gives you a chance to intervene in a way that maybe genetics doesn't always suggest directly. So let me talk about personalized nutrition in the real world. After this successful Pioneer 100 study, we decided to launch a company. So Lee Hood and I, are, along with Clayton Lewis, are co-founders of this company called Airvail. It delivers scientific wellness, so it makes a lot of the same kind of measures that I just mentioned, and teams with coaches and so forth. We have now about 175 employees at Airvail, and we've raised about $54 million for this company so far, and we've got thousands of people now coming through that program. In a nice way, Airvail and ISB actually have a partnership so that for people that go through Airvail, if they check a box and say, yes, my data can be used anonymously for research so that we can learn more about health and disease to help with their health, their kids and their grandkids. For all those people that say yes to that, we actually get access to de-identified data for scientific discovery. So this is bringing in now thousands of individuals through this program. We're very, very grateful for that. In terms of the value proposition back to an individual, it's really about empowering them with data, coaching to help interface this with the behavior in their life so they can take action, which yields results. And we now have, or Airvail has, just tons and tons of stories, which if I had more time, I would go into. There's been very interesting cases, not only of people who do the usual things like lose weight and get healthier, but actually there's been a number of cases where some of these, even though we can't directly connect them causally, we've had a number of people come through where we've been able to direct them into their physicians for early warning signs. We had one woman actually in the last month where, I won't get into the details because I'm short on time, but where we were able to get an early warning sign that led to the discovery of a stage 3 colon cancer that was large enough to almost occlude the entirety of her colon, that was cut out by the doctor's estimation just immediately before it was likely to go metastatic. And so that was an incredible one just from the last couple of weeks, and there's many, many of those kind of cases. So there's technology as well to try to amplify that coaching relationship through mobile apps and dashboards and connections. So I have almost daily texts with my coach, as do many people, in terms of diet, nutrition, and things like this. And so this is a program that we hope people will stay with over the course of their life that will have a huge impact on their health. So that's really the goal with something like this. And so lastly, I want to just share a little bit about another company, just to give you a sense for what else is going on in this space, which is a company called Habit, which was spun out of Campbell's Soup as the parent company. And this is really a great company as well. It was started by Neil Grimmer, who is a terrific CEO there. And basically what they do is they take a lot of information. They consider themselves kind of the world's first personalized nutrition company. There's probably others that would claim that mantle as well. But basically here they get some genetic variants. So this isn't like whole genome sequencing or anything, but they measure certain variants, certain measurements out of the blood. So it's, you're talking about, a fairly reasonable kind of set of markers, 40 or 50 different kinds of markers that are measured here. They also do a challenge shake, which people take, and then you get blood measurements at 4 time points. And this essentially looks at the dynamics of processing carbohydrates, protein, and lipids. You put in a little bit on habit and goals, and essentially they run this through an engine. And essentially what happens is you have testing, you get your results, coaching, and then importantly, and what's very much a differentiator here, is that at least in certain markets, they will then deliver you personalized food. And I think this is really an essential part, because that behavior change or really integrating with what is, all this information is tough. And what habit actually does is provide food back to individuals based on, so if it said you needed to eat more, of X, Y, and Z, if you need more omega-3s or more vitamin D, or whatever, or vitamin E or things like this, it would actually shift your requirements in that way, which I think is really great. So to sum up, basically, what I've talked about is that we are starting to be able to gather immensely more kinds of data, these personal dense dynamic data clouds. These kinds of concepts are already being put into practice through companies. I talked about just a couple of those. I really think these kind of data can be the foundation for the future of personalized nutrition. So with that, I'll just thank everyone that was involved in the study and various members of our lab at ISB. Thanks.
Thank you, Nathan. And I'm sure all of you are saving your questions for our discussion session at the end of this period.